Hey guys, welcome back to my channel. This time we'll be talking about exercise 49 or urinalysis. This will also be a part of your finals together with the urinary system. So I hope that you'll be able to listen to this discussion and be able to study it appropriately. If you're ready, let's start. So as mentioned, urine is formed in the kidneys and it goes through a three-part series of process from filtration to reabsorption to secretion. After it goes out of the body, the collection of urine actually gives us a good insight into what is happening inside. So the urinalysis is an in-depth assessment of the renal status using an easily obtained specimen, which is urine. It is a quick indicator of an individual's glucose, protein, ketone status, and also the function of the liver. You might be surprised, what? The urine gives us a clue of what's happening in the liver? Yeah, later on we'll go to that. So the process of urinalysis is divided into three. First is macroscopic. This is that picture in the middle. Macroscopic meaning we'll have to observe the physical properties of urine first. Color, odor, even the volume at times. We also consider that. Next is microscopic. This is the one that you see at the end. So we put urine sediments in a glass slide and looked at it under the microscope to see the microscopic elements found in the urine. And lastly, we have the chemical, which is shown in this first part of that image. So we also try to check what chemicals are found in a patient's urine. So for the macroscopic analysis of urine, we take note of the following. We take note of turbidity, meaning the clearness of the urine. Because when it is too turbid or when the urine appears cloudy, then it may give us a clue that maybe there is an infection, maybe there is contamination, or maybe there is dehydration in the patient, anything that can make the urine turbid or cloudy. We also check for the physical appearance, mainly the color of the urine. So normal urine would be appearing as clear to watery, similar color to water, or yellowish. So what gives the yellow color to urine? It is your urobilin. Urobilin. Okay? And this urobilin is an end product of an analyte formed in the liver. Okay? So when there is a liver problem, urine might appear pale. They might lose that normal yellowish color. But not all clear urine are abnormal. Okay? We also try to check for odor. And we do not smell urine directly. We use a whiff test. So we try to manipulate the air, so that we can smell the odor. And lastly, volume, as mentioned. So polyuria is what we call increased urine volume. Okay? And it is observed in patients with diabetes, whether it be diabetes mellitus or insipidus. So when there is too much urine being formed or when you go to the CR too often, then you might want to have your sugar checked because that might be a symptom of your diabetes. So that's for your macroscopic, and we take note of that. And if you do have a urinary result, you might want to check the macroscopic results of your urine. Next, we have the chemical test. So for that, we used a dipstick. Okay? So it is a strip with multiple panels, as you can see. And each of that panel has chemicals which will react with the chemicals found in the urine. So we will put it there and then compare it to a standard which is usually found in the bottle. So this is how it looks like, We hold the strip against this reading, and then that's where we get the values. Okay, kung negative ba yung leukocytes, or is there trace, is there moderate, large amounts? So we base it on the color that will be produced by the test strip. So what are those 10 chemicals that we look for in the urine? Those that are included are your leukocytes or their WBC as a marker of infection. Nitrite is also an indicator of bacteria. So when there is UTI, we usually expect positive leukocytes and positive nitrites. Your urobilin, as mentioned, no? It is an indication of RBC destruction or liver disease. You also have your proteins, which also indicates kidney disease. We also have blood, kasi if there is blood in the urine, then there might be kidney stones, there might be an infection, or there might be a serious kidney disease, no? Because blood should not be seen in the urine since it's only the filtrate that is observed, no? Remember? Also, the specific gravity. It gives us a clue as to the hydration status. Ketone is a byproduct of fat metabolism. Bilirubin also for liver disease. And glucose. Because when there is glucose in the urine, then meaning there is too much glucose in the blood. Because urine is formed from the blood. And lastly, we have the microscopic examination. So, urine is subjected first to centrifugation to gather the sediments or to allow the sediments to settle at the bottom of the sample. We then remove the supernatant and we get the filtrate or the sediments and we put it on a glass slide and examine it under high power objective. Under the HPO, you will observe substances such as your crystals, your white blood cells, of course, bacteria because there's normal bacteria in the urine, casts, artifacts, and even semen. So actually, a more extensive discussion on urinalysis will be taught during your third year. But it will be helpful to start being familiar with it this early on. Sometimes even parasites can be found in your urine, no? And you'll go to that also during your third year, your parasitology. These are the different microscopic structures that you might see. You have your normal crystals and you also have abnormal crystals. And this is how they look like. They are called crystals because under the microscope, they take on a very shiny, very unique looking, sometimes even geometrical, no? Kasi ganun yung mga crystals, diba? They appear geometrical. So that's how they look like also under the microscope. So that's it. No, that's it. Please do answer that lab report. No lab reports needed for urinalysis. Thank you and God bless.